Okay, so I'm Dan Rabosky from the University of Michigan, and I'm going to talk today about some comparative ecological work that we're doing looking at some of the world's most diverse squamate communities. Specifically, we'll get into looking at some patterns between arid Australia, and the lowland Amazon, which are two of the most diverse places on Earth for squamates. And before I begin, I just want to thank the organizers of the conference, and especially Felipe for bringing me here and for all the hard work in organizing this conference. So it's been a fabulous time, and I really appreciate the work that went into organizing things and making this happen. Can everyone hear me in the back, by the way? Raise your hand if you cannot hear me. So you cannot, okay. Do I need to speak louder or closer to the microphone? More? Louder, microphone more. How about this? Is this a little bit better? All right. So what's really motivating my talk today is this pattern of global richness of squamate reptiles. So what you're seeing here is a heat map of squamate species richness in a global sense. And what you can really see from this map are that a couple of places really stand out as having the highest regional diversity of squamates. And on one hand, you have the neotropics, especially the western Amazon basin, as a region that is characterized by exceptionally high richness of squamates. And another region that stands out, especially for lizards, is arid Australia, and that's been famous for a long time, based on work from Eric Pianka. And really, the neotropics are the center of global snake diversity. And Australia is basically the center of global lizard diversity. I'll try walking around this thing. And Australia is really a center of global lizard diversity. And so what you're seeing there on the right is really a map of it's breaking that species richness map down into the regions of the planet that have the highest concentration of snake species richness, basically the richest 5% of geographic regions on Earth. And on the other side, on the left, you're basically seeing the map broken down by regions that have the highest concentration of global lizard diversity. And so what you can see is that the neotropics really light up for snakes, and Australia really lights up for non-snake squamates. And I sort of use, even though lizards are obviously not exclusive of snakes, they're not monophyletic, I'll nonetheless use that distinction between lizards and snakes throughout my talk. And so what I'm going to do is going to be some pretty simple things. I'm going to focus first I'm just going to contrast some basic aspects of the ecology of these systems and hopefully give you a little bit of a sense of what it's like to actually work in some of these systems. I realize a lot of folks here have probably done a lot of work in lowland tropical forests, but Australia, and arid Australia in particular, is quite different in a lot of ways. And then I'm going to come back to, after summarizing a few patterns, I'm going to talk about some key questions that we can't answer that I find very fascinating kind of going forward. So for the next few slides, I'm going to introduce you to some aspects of the setting and cover just a little bit about what it's like to work in arid Australia and the Peruvian Amazon. And so this is Australia. And so the Australian arid zone, which is basically what I'm focusing on here, is a region that is really circumscribed by this yellow line. So this is basically the Australian desert. So about 70% of Australia is arid. And now within that arid zone, Australia has long been famous for having really, really high species richness of lizards in sympatry. And so if you go to Australian spinifex deserts from the western part of the Australian arid zone, you could find up to 70 or so species of actual lizards living together at a relatively fine spatial scale. That's basically like a 1 square kilometer patch. You could have 70 species of lizards or more in sympatry at that scale. So this is sort of an unparalleled level of diversity, and it's not driven, and that diversity is not biogeographic diversity. It's actually, these are like actual species that are basically in just one habitat patch and the next habitat patch. So it's incredible, and it's incredibly rich compared to other desert systems in particular. So if you go to the Kalahari, maybe you find 20 or so species in sympatry. And if you go to the richest deserts in North America, at that sort of spatial scale of a 1 kilometer by 1 kilometer sort of scale, you're going to find very few places, if any, where you could find more than 10 species of lizards in sympatry. So Australia really stands out. And these are places that I've worked around the Australian arid zone since 2004. They're basically spanning a lot of the continent in this sense. And Really today, though, I'm going to focus on one specific community that we've studied most intensively, and this is a place called Lorna Glen, or Mattawa is its indigenous name. And so this has been an aerial view of Mattawa. So it's basically essentially an old sheep station. So it's a fazenda or a finca, and it's very, very isolated. And for the last 20 or so years, it's basically been managed as a conservation reserve by the Australian government. And this is just an aerial view of the old sort of farm quarters. And one thing about working in arid Australia, so Australia is about the same size as the United States, same size approximately as Brazil or China, yet it has about 21 million people. And on top of that, almost all of those people are stuck in about 6 cities on the coast. So Australia is the country that has the lowest effective population density on Earth. And when you get into the center of Australia, you see things like this, where all the tracks basically look like this, all four-wheel drive tracks. And from a track like this, for example, you basically have distances like this to the nearest paved road. So from this track right now, where I'm standing on top of a Land Cruiser taking this picture, you can go 200 kilometers one direction before you hit the nearest paved road, or 1,000 kilometers in the other direction before you hit the nearest paved road, or about 600 kilometers straight ahead before you hit the nearest paved road. So that's what the interior of Australia is like. So very, very remote, very empty, and so on. And just to give you a sense of what some of the habitats look like within arid Australia, so this is a spinifex sandplain. And so that low sort of hummocky vegetation in the foreground is known as spinifex. And this is basically a really prickly, really prickly, but not like woody spine sort of vegetation that basically provides really good shelter and cover for lizards. And in the background, you sort of see this very, sparse, spotty overstory of eucalyptus trees. And this is really so it's almost like a savanna, even though this is quite arid. And spinifex is basically something, you can think about it kind of as a coral reef for lizards, if you want to think about it like that. So this is the habitat this specific habitat right here is the habitat that has the highest rich alpha diversity of lizards on Earth. But there's a lot of other landscapes and other types of habitats that are mixed in within the Australian desert. So it's very much a mosaic of different habitat types. So we see those types of spinifex sand plains that we see will be interspersed with sand dunes, just very long parallel ridges of sand. Sand dunes are very widespread, and they're interspersed with other habitat types. So it's very much a mosaic. The entire landscape is extremely flat. There's occasionally very low, rugged ranges that basically aren't any higher than a few hundred meters at most. And so most of the land, there's essentially no rivers that drain out of the entire Australian arid zone. So that's something that's kind of surprising. There are no permanent bodies of water that flow, and there are no rivers that flow out of it. So everything basically just pools in a set of very low depressions scattered all across the arid zone. And you basically get habitats like this that form throughout that. So this is a saltbush lake with halophytic vegetation, and that's a very common type of vegetation. And there are species of lizards that are specialized basically for all of these different types of habitats. And then you also have sort of erosional surfaces like this, and these landscapes are very old, but basically you have these places where you're just, you're not sand dune, but you're kind of have a landscape that's just the eroded rocky rubble of things that have sort of accumulated, the detritus that it's accumulated in place for literally millions of years. And often the vegetation there is associated with things like acacia trees, like you're seeing right here. So we kind of have a rocky, rubbly surface and acacia savanna in the background. Now, if you look at an aerial view of this is sort of what it looks like. This is a photo I took from a helicopter flying over this. And you can see this sort of landscape mosaic here. I'll step away from the mic. But this kind of habitat right here, this is sort of this spinifex sandplain patch right here. You can sort of see hints of sand dunes right here. This is acacia savanna back here, and so on. And these lighter patches of vegetation back here are spinifex sandplains again. And so the whole sort of setting is very much one of a mosaic of different habitat types. And how do we sample these kinds of communities? Well, just like you would do here and maybe some of the habitats like Restinga or Cerrado, we use pitfall trapping very heavily in Australia. So it's a pitfall line going in. And so imagine many folks here have done pitfall trapping. This is a pitfall line on some dune habitats. And another thing that's kind of an ecologically interesting aspect of the Australian deserts is that this is very much a high disturbance, fire-driven landscape. And so spinifex is very flammable. So this is actually has a number of resins in it that actually cause it to burn at a very high rate. And so if you were to look at this is the aftermath of a fire. So stuff will regenerate over the space of a few years. You'll start to get little spinifex bits coming back that'll sort of look like that, and then eventually over this maybe 15 to 20 year time scale, you'll basically get full-on spinifex hummocks, which will be about this high, like that. And that's sort of like prime lizard habitat. But there are actually species of lizards that specialize on these early successional fire landscapes as well. And so if you were to look at an aerial photograph or look at Google Earth imagery of the Australian deserts, especially the spinifex deserts from the western Australian arid zone, this is what you will typically see. And what you're seeing here is, and what you will see if you start looking at this kind of imagery, is very much a landscape that is driven by fire. And so the darker landscape that you're seeing here is basically mature spinifex sandplain, and the cleared areas are basically fire scars. And so what you'll see is often very straight lines associated with those fire scars. And so basically the fire just moves along with the wind. And so you'll have a fire that starts here and blows down like this, and then maybe you get the wind will switch and you'll get little, there's probably another fire right here, little secondary fires going like this, and so on. So fire is very common. Fire is very heavily has shaped this landscape and continues to shape this landscape. And it's very, very evident even from looking at imagery for the desert regions themselves from Google Earth or whatever. This is just a little analysis from a paper that was published that looked at the distribution of burn sizes for Australian bushfires. And one of the things to notice, though, from this, the only thing is that there's actually, on the x-axis, that's the log of the size of a particular burn in kilometers squared. So one of the things to notice is that while there's a fair number of small burns, there's actually a lot of really big burns. So you have burns that are every year that are exceeding 1,000, 10,000 square kilometers, and about 2 to 5% of the landscape burns every year. And so our data that the stuff that I'll talk about are based on is the result of about 180 days of sampling distributed over about 7 years of work, different seasons. So the typical pitfall sampling period is about 7 to 8 day units, and we hit this in the winter, spring, summer, and fall. And a lot of this is all in collaboration with a long-term Australian collaborator of mine, Mark Cowan. And so we have about 6,000 squamate records, which seemed like a lot. And then I was sitting this morning through a talk about Restinga work, and it was like 70,000 records. So yes, this is a small fraction of that. Nonetheless, it's what we have to work with. Now, contrast that with the other site I'm going to talk about. So this is now moving to the Amazon, and this is basically Greater Amazonia. And I'm going to talk about work we've been doing for the last 3 years at a research station in the Peruvian Amazon called Los Amigos. Now, the interesting thing about these, and I guess one of the reasons what sort of motivates my interest in this is that, as someone who mostly does, I think about macroecological and macroevolutionary patterns, broad-scale patterns of species diversity. And I think a lot of times in macroevolution or macroecology, species richness is just kind of this variable that sits there like, oh, we're just explaining species diversity. We have like bird species diversity is this here and this here. So what we're trying to do here is say, okay, well, squamate richness more or less similar between these regions, but let's actually dig in and see how these systems really differ in terms of their fundamental ecological attributes. And so this is just some of the kinds of landscapes we would encounter in the western Amazon basin. And this is work that's been in collaboration with my postdoctoral researcher, Rudy Van May, who's been working in this area since 2003. And so he's accumulated a lot of data, actually, especially on frogs, even before he started working with me in 2015. But so we've run a number of trips to Peru now. So these are some of our field teams over the past 3 years. We have a lot of Peruvian collaborators who come along as well. Occasionally we teach field courses for Peruvian students and herpetologists when we're there on things like scientific data analysis or computer programming and quantitative methods. And just give you a sense of what some of the landscapes here look like, especially to compare this to what I've said for Australia. And if you look at satellite imagery for the western Amazon, you can't see any of this same level of heterogeneity. but you do have some different things. We have terra firme and floodplain forests, kind of the classic shift between plant communities in particular. And then you've got riparian stuff and palm swamps, for example. But it's not quite the same as on the same scale that you would have in Australia. And let's just show you what some of these habitats look like. Palm swamp, bamboo thickets. So bamboo is actually quite common in the southeastern Peru, in the southern Peruvian Amazon in particular. It's common everywhere, but it's especially common there. It forms fairly large tracts. And then there's obviously terra firme and floodplain forest as well. Now, just to contrast some of the patterns in Australia with the patterns in the Amazon, I'll say a couple things about disturbance here. So this is really a system that is driven by much smaller scale disturbance. So here we're looking at a system that's basically driven by tree fall gaps. And the tree fall gaps obviously can involve multiple trees because, tropical trees are basically tangled with lots of other things. So when one big sort of emergent tree comes down, it might take out 5 or so other trees in the neighborhood. But nonetheless, the scale is always pretty small, and it's pretty consistently small from that perspective. So it's a much smaller tree. So this disturbance is frequent, but small scale. And when we're working here in the lowland Peruvian Amazon, we're using a combination of pitfall traps and funnel traps. So these are funnel traps and pitfall traps again. And funnel traps are actually quite effective for some kinds of things that we basically can't get in pitfall traps, especially snakes. And so this is also paired with extensive active searching. So if you've worked in lowland tropical forests, you know that a common way to find reptiles and amphibians is basically go out at night with a headlamp and look for things. And you sit there and stare and stare and stare and walk around and look at patches of leaf litter like this until something turns into like a ceratophrys. So this is just what you're spending hours every night doing. Actually, I spend a lot of time staring at green things, hoping that sooner or later something will actually turn into a snake. Very rarely we do find snakes. Let's try Maluber. And so overall, we've got about 1,800 squamate records, and that's from 2003 to 2019. That's using a combination of standardized pitfall trapping and funnel trapping, as well as litter and transect surveys, and some just sort of opportunistic visual encounter type surveys at night. Now, and one of the goals of this work is really to not just sort of compare and contrast the ecology of these systems, or to answer some macroevolutionary or macroecological question that, when we go to the Amazon, we're going to collect the data for this. Really, we're trying to contribute to this sort of global knowledge of basic biology and natural history of reptiles and amphibians in lowland tropical forest. And so when you look at a Bothrops bilineatus like this, we might ask a lot of questions about this, like, what habitats does it use? How does its trophic niche differ between populations? How abundant is it? How does its abundance change through time? How is its population structured in space? How does this species interact with predators, parasites, and prey? And how does it behave, and how does its behavior relate to those other facets of its natural history? And so we're really just trying to document basic natural history as well. And as a curator in the Museum of Zoology at the University of Michigan, a lot of what I think about is how we can take these kinds of data and make them into a permanent record that people can access, either in the form of vouchered specimens. So we collect specimens very heavily wherever we work, whether it's Australia, like you see on the left, a bunch of aranid specimens, or in Peru, which you see on the right. And we try to take as many things as we can. We think about specimens essentially as super specimens that have a number of things associated with them, not just a tissue sample and a voucher, but we also take multiple tissue types that we can use for genetic purposes. We also take things for doing transcriptomics. We take skin swabs, secretions, fecal samples for doing other sorts of downstream genetic and pathology analyses. We take things for chemical ecology, so we take skin sections from frogs and preserve them in methanol for basically doing skin toxin analyses. And we take venom glands and sometimes venoms themselves. And then we take quantitative habitat and activity data, and all this stuff basically ends up associated with the specimens in our collections at Michigan. And in addition to collecting basic data on, that's associated with a specimen as sort of a focal point like that, we're spending a lot of time out there. So, when you spend a lot of time out places looking for things, you document other facets of their natural history as well. So we're keeping an eye open for, interesting predator-prey types of observations, including not only cases where squamates themselves are the predators, but also cases where squamates are the prey. So this is a tenid spider eating a cercosaura. This is some data that some results, observations that we summarized and published earlier this year. And sometimes when you're out there looking around, you find some really mind-blowing things. So on the next slide, I'm going to show you some video that a couple of my graduate students shot a couple of years ago on one of these trips. And they came across, I heard a little kicking in the leaf litter, and what they came across was a very large tarantula, so a theraphosid spider preying on a Marmosops mouse opossum. So this opossum, not counting his tail, is about this long. And this tarantula is actually dragging it off. So they heard the scuffling, basically the death kick of the actual opossum. And then they watched this tarantula for about 5 minutes, actually, as the possum essentially became completely paralyzed and the tarantula moved off with it. So every now and then you actually find really, if you work especially in tropical forests, you find really mind-blowing things like that. Very infrequently, but... One of the other things we do is we try to collect standardized behavioral data. And so this is a Mycurus sornamensis, filming it immediately after catching it. And you can see it's basically got that stereotype Mycurus type defensive display. So we actually do this with almost every snake that we catch, at least until we have a very good representative sample. And we do this in a number of different ways, and we have some standardized behavioral protocols that we use in the field for catching this kind of data. And a lot of this work is actually, even though this stuff will ultimately all be publicly available and associated with the actual museum specimens, this is motivated a lot by my wife and co-curator of herpetology at the University of Michigan's research interests on evolution of warning coloration and mimicry in snakes. And so the kind of things that we do in the field is we create these standardized behavioral arenas like this. You have a Mycurus obscurus in an arena with a set of cameras that's actually going to film the behavioral responses to a number of sorts of experimental treatments that we do. We do these within usually a day of catching a particular snake, and we take videos immediately upon encountering snakes in the field. And so ultimately the point here is to try to understand things like why on the left you have this Mycurus obscurus showing a stereotyped tail-raising and flattening behavior, and how you get snakes like this Atractus elaps, which is a completely harmless coral snake mimic with a nonetheless very coral snake-like display, how you have the evolution of those kinds of behaviors. Now, one of the cool things about this, though, has not only been, I've learned a lot about snake behavior just taking these kinds of data, but one of the cool things about this is not only seeing the coral snakes do and what their potential mimics do, but also what some of the other snakes that we see do in the field. So, for example, here's a Xenodon severus. So you can see on the right, you can see these nice rear fangs. This thing's actually reacting to one of the experimental behavioral treatments. And a Xenodon basically acts kind of like a cobra. It's hooding, it's gaping, it's actually tracking the person or the bag that's actually going over its head. And so this is a fairly threatening snake in a lot of ways. So at least it thinks it's threatening. So just to summarize some of the ecological attributes of these systems. So that's just a little bit about a snapshot of what we do in the field. So Australia, you could think about it as it's red, there are a few trees, and it's dry. So these are like really simple ecological differences between these systems. The Amazon is wet, it's green, there's lots of trees. But there's some similarities between them. So you could also say that here in the Amazon, or in Australia, we have a system with extreme diversity. It's a vast area, the Australian arid zone. It's a patchy landscape with lots of heterogeneity. It has frequent disturbance, and the scales of those disturbances can be very large when an area of 10,000 or more square kilometers actually burns, spinifex landscape. Now, in contrast, you can look at the Amazon, and here we have a landscape with extreme diversity, also a vast area, but it's also a relatively smooth landscape, and it's mostly terra firma or floodplain forest, and that interface between those can be a fairly fine grain. And we have frequent disturbances, and the scale of those disturbances is pretty small. And just to look at some of the major clades that we'll find in these systems. So this is a phylogeny that's been pruned from the Tonini et al. Phylogeny from a few years ago, and we've added some other taxa to this tree to basically have a complete phylogeny for all of the squamates from Los Amigos in Peru and Lorna Glen in Australia. And I'm just going to show you a little bit about these clades just to give you a sense of how these communities might differ phylogenetically. So when you see something in red, that means Australia, and when you see something in blue, like tips in this tree, that means Peru or the Amazon. And so if you're going to contrast these systems in terms of, say, geckos, we have geckos in both systems. So if we look at Peru, we have 3 species of geckos in sympatry in the Amazon. 2 examples here, a Gonatodes and a Pseudogonatodes species, and there's also a larger Thecodactylus. And that's basically it for geckos. However, go to Australia, we have 17 species in sympatry at the 1 kilometer square scale, including things that are very typical geckos that you might think of, like this Gahyra variegata, probably one of the most common geckos in Australia. And you can see the nice toe pads there because it's arboreal. However, the core of Australian gecko diversity is actually in several other radiations. So the diplodactyline geckos in particular, which are groups, which in the case of the genus Diplodactylus, which is quite diverse, is essentially terrestrial. It doesn't have toe pads. And you also have knobtail geckos in that general group. Other knobtailed geckos and so on. So gecko diversity is extremely high, and some very charismatic and cool geckos out there. Now, there's also clades in both Australia and the Amazon that are, and I'm thinking now in terms of ecological attributes of these clades. So if you look up at the top here, these are clades that are active foraging, use their jaws to catch prey, and they tend to have well-developed chemical prey detection. And so this is clades that traditionally, within more traditional squamate phylogenetics, would have been thought of as scleroglossan clades. And so within the neotropics, for example, we have things that, we have skinks, we have gymnothalmas, and we have tias, which tend to fill those roles. A couple examples of those there. But in Australia, within skinks alone, we have 25 species of skinks in sympatry versus, roughly 2 to 3 species in the western Amazon. So, and a lot of that diversity is actually concentrated disproportionately phylogenetically in a few groups that are major contributors to overall diversity. This is a Tenotus. There's actually 10 to 14 species of Tenotus in sort of regional sympatry at that 1 kilometer square scale that I'm referring to. There's an assemblage of set of Tenotuses that you could find pretty easily at Lorna Glen in Australia. And you also have some more larger, some might think of them as more charismatic skinks, like blue-tongued skinks like Teliqua, which, including the tail, can be up to about this long. And they have very nice defensive displays like this guy right here. Now, can't talk about active foraging clades in Australia without mentioning varanid lizards as well. So, of course, varanids are major players with respect to the ecology of Australian squamate communities. And amazingly, there's actually 7 species of varanids in sympatry that you could find even at a single site within this region of the Australian arid zone. They range in size from these tiny little pygmy short-tailed goannas, like this Varanus brevicata that you're seeing right here. This is an adult, a large adult actually. This is basically a spinifex specialist, basically spends most of its life in spinifex clumps. You'll basically never see this lizard out, but it's readily pit trappable. And then we have some more intermediate-sized varanids, including Varanus aremius and Varanus panoptes, and panoptes will get up to about this big. And then at the largest end of the spectrum, we have Australia's largest lizard, the Parentes. So these will be, including the tail, will be over 2 meters long. So this is a very large lizard. They have a full size range of varanids, and they're relatively evenly spaced in terms of body sizes within a local community. Now, switching from active foraging, chemosensory, jaw-capturing lizard clades to the other major ecological types, I'm thinking about traditionally we often think about lizards as falling into 2 major ecological categories, one of which is active foraging and the other of which is essentially sit and wait. Now if we look at how sit and wait clades actually species differ between these systems or are similar between these systems, we have a lot of parallels. So in the neotropics, we have anoles and tropidurines, iguanians more generally. And in Australia, we have a clade known as agametes. And in a lot of ways, this genus Ctenophorus and several other genera of Australian agamas are very similar to typical tropidurian type lizards like Tropidurus or Stenocircus or Plica. But there are many other groups of agamids, including things that you may be familiar with, but they're totally bizarre and don't necessarily have a clear analog in the Amazon, like the thorny devil, Moloch horridus. And you may have seen lots of photos of Moloch, maybe have not seen photos of baby Molochs. And other forms like pebble dragons right here. So this is a Tympanocryptus cephalus. So this is a species that's an almost perfect pebble mimic down to the level of its tail looking like a piece of dried vegetation. So now on to snakes. So Australia has about, the Australian community that we work in at Lorna Glen has about 10 sympatric species of elapid snakes. And so these include perhaps the most famous deadly snakes, brown snakes, and mulga snakes or king brown snakes, the genera Pseudonaga and Pseudechis. But they also include a lot of other things, including Australia's viper. So it's not really a viper, but it's a death adder, which is a very structurally similar snake in terms of ecology to New World vipers. And a number of species of essentially small fossorial or semi-fossorial elapid snakes that are completely harmless, like shovel-snouted snakes like Brachiophis, like you're seeing right here, or Samoselaps, which you're seeing right here. And there are places where there are 5 or 6 species of these things in sympatry in the Australian arid zone, these species of small, fossorial, harmless elapid snakes. Now, that diversity of snakes, though, in Australia doesn't actually hold a candle to the neotropics. That's actually one of the reasons I'm excited to be working in the neotropics, is because I'm very excited about snakes. And the neotropics really have unparalleled snake diversity. It's like nowhere else on Earth. And so obviously you have things like boas and pythons, pit vipers and coral snakes, but we also have this insane radiation of dipsodine snakes, 700 or more species of snakes, mostly neotropical. Definitely one of the unsung sort of, macroevolutionary success stories that most people outside of herpetology and neotropical herpetology don't ever think about. And yet this is a massive evolutionary radiation in terms of ecological space and species diversity. And so I find this radiation of dipsodines to be quite fascinating. So the blue arrow is indicating where dipsodines, basically the fraction of that community that basically dipsodines. So what we basically got is a system where we have 2 communities, and they're both drawn from a fairly diverse set of clades overall. So you got Australia in red, Amazon in blue. And what you're going to see on the next slide is basically the species richnesses within those local communities of all of those clades, just a rank order plot. And so the point of this is just showing that the overall shape of these things is basically the same. If you're trying to think about squamate clades in general, you don't really have a system where the Amazon is dominated by almost exclusively one clade and there's lots of clades in Australia. In general, you have a number of clades that have relatively high diversity in both systems, and then a fair number of clades that have relatively low diversity in both systems. So there's a lot of similarities between these overall distributions of clade richness. And the richest clade in the neotropical communities in the Amazon are the dipsodines, and in Australia, it's the sphenomorphine skinks. And when I say clade in this context, I'm really talking about independent evolutionary radiation. So if you want to think about these as each of these as like a potentially separate colonization of each region, whether it's the neotropics or Australia. And in terms of comparing the ecology of these systems, I'm going to focus on 2 fairly simple attributes. And first I'm going to talk about their abundance and diversity structure, and then I'll talk about trophic ecology. So let's look at the species abundance distributions. This is just based on the data that we have for these communities, and we'll just look at each data point you're going to see here is going to be a species plotted by its abundance and ranked by its abundance. And so we're just going to look at the actual shapes of these species abundance distributions. So broadly speaking, these species abundance distributions are comparable. They kind of look the same as species abundance distributions for many other complex communities. You have a couple of pretty common species. You have lots of relatively rare species. However, and this is a huge difference that I alluded to earlier, the neotropical communities are about 70% snake. So a local community in the neotropics is about 70% snake versus about 17% snake in Australia. And that includes not only elapids, but there's a few species of blind snakes in there in Australia and a couple of species of pythons as well. But nonetheless, about 17% snakes. So a vast, huge difference in terms of what snakes have actually done in these two systems. And most species in the neotropical communities are snakes, and snakes are both species poor and uncommon because they also fall at the rare end of that spectrum in Australia. So this is just a simple rarefaction plot, just kind of getting at how species-rich is the Amazonian community and the Australian community on a sort of per capita basis. And you can see that the Amazon is a little bit more diverse on a per capita basis as you'd measure it with rarefaction. I don't make too big of a deal about this particular plot right here. But what I'm going to show you here is basically another way of viewing that rarefaction curve. And so here I'm going to say, okay, we have fewer records in the Amazon than we do for Australia. But what if we standardize the Australian data to the same level as the Amazon data, and we ask the simple question of first of all, how many species in our Amazon data set are represented by just one individual per species, and how many would we expect in Australia to just have one species as well? So in Amazon, we have 14 species in that data set that are singletons. One individual that we've known from for more than 10 years of field work, actually. Now in Australia, standardizing for the amount of data, that's less than half of that number. So we would have, at the same number of records, correcting for that, we'd have just 4 or 5 individuals that we would expect to be known from a single specimen. And if we just asked how many rare species, that's like 1 to 5 records would we expect, again, it's about 50%. And so the take-home message here is that per capita, the Amazon has roughly twice the number of rare species as Australia. So there is a difference here. This is our species richness curve for our first University of Michigan expedition to Los Amigos in Peru. Just looking at our cumulative species accumulation curve just for, on a per day basis, combining different methods of sampling the community. So this is 21 days with about roughly 10 people each day doing stuff. So, species richness kind of builds up. It's getting nowhere near an asymptote here, by the way. It's going to keep rising. But you can contrast that to the first 7 days I worked in the Great Victoria Desert. So here I'm a first-year PhD student. I show up at the Great Victoria Desert at this field site working with my collaborator, Mark Cowan, and this is our species accumulation curve for 7 days of pitfall trapping in Australia. So we basically catch 40 species on the first day, and you add to that every day. So this is unlike anywhere else I've ever seen on Earth. So if you were to give me 2 weeks of pitfall trapping in arid Australia, in spinifex deserts, in say probably October, November, 2 weeks would be enough to get 95% of the community across the board. It's that pitfall trappable, or that species are that detectable in that sense. Here's another view of this same phenomenon. So this is the number of different species of squamates we've caught per day, pulling together results from here. We got 4 different trips to Peru, looking at our active days where we have traps in the ground and everything. So you can see the average number of species per day is probably hovering around 5 or 6 or 7. And this is the equivalent view of species per day for the Australian fieldwork. So you have days where you're getting, maybe the average is in the 20s, but you have days where you're getting up to 40 species. Actually, the Australian stuff is only pitfall trap records as well. If I count site records on that as well, that would add a few more species. So there are single days in Australia where with a reasonable sized grid of pitfall traps that you can run with a team of 3 or 4 people, you will catch 300 individual lizards. So things are very trappable in Australia. So just summarize a few of these properties of diversity and sampling. So there's more rare species in the Amazon, and encounter rates and trapping success are higher in Australia. I haven't shown you the details of per bucket pitfall success rates and so on, but the basic story is that there's more rare species in the Amazon. That holds no matter what, no matter how you look at the data, and encounter rates and trapping success are higher in Australia. Just a better place for pitfall trapping, essentially. Species are more detectable. And what it means is that we have very different amounts of time and effort that we need to understand the ecology of these communities. So now I'm going to talk about the trophic ecology of these systems. So we've got, okay, broadly comparable patterns of species diversity, broadly comparable clade structures across these things in the sense of there are some, there's roughly the same total numbers of clades that make up those communities. And so what we've been doing is tabulating dietary data for as many species of squamates from these communities as we can, including stuff from published works like this right here, including Laura Alencar's paper on pseudoboine snakes, but many, many others. So this is really work that includes published observations from things like Eric Pianka's work on the natural history of Australian lizards, some unpublished data from Laurie Vitt, and a number of new records that we have, and field observations from my graduate student, Mike Grundler in particular, as well as compiling literally hundreds of articles that have to do with snake dietary records. And so Mike has gone through and literally compiled thousands of records at this point from multiple natural history journals of snake dietary records. And so just looking at what we have for Australia and the Amazon, we have this data set where we've scored all these things into about 28 diet categories. This is about 1,300 snake prey records from the species that we have here. This is 69 Australian and 65 Peruvian species that we have dietary data for in some form. And what I'm going to show you here is basically just a simple multidimensional scaling analysis just to ordinate those species by diet category, just to see if we can see some patterns breaking out between the Australia and the Amazon. And so, again, you've got red points, Australia, blue points are the Amazon. And you can see a clear split in some ways between where blue points are concentrated and where red points are concentrated. And I'm not breaking this, this is snakes and lizards together. Now, these NMDS axes actually correlate really nicely with some major breaks in trophic ecology. So that gray line that you're seeing right there now is basically the split between things that mostly eat vertebrates and things that mostly eat invertebrates. And so if you code things by snakes and lizard categories, for example, you can immediately see that there's a big split where most snakes tend to eat vertebrate prey and most lizards tend to eat invertebrate prey, with a few exceptions that you can see kind of spilling over into different into the other side. So that's a big key or clue into trophic differences between those systems right there. Now we can also look at this, break this down a little further. We see that these NMDS categories essentially also track nicely some major types of vertebrate prey and invertebrate prey. But in blue, you've got basically things that mostly eat squamates. In red, you've got things that mostly eat endothermic vertebrates. And in green, you've got things that mostly eat fishes and amphibians, or wet vertebrates, essentially. And you've also got sort of the oddball snakes, especially in the neotropics, that eat other sorts of dietary things. So you've got things like Tantilla and Dipsas, which generally tend to be invertebrous, eating things like mollusks and centipedes, and things like Atractus, which tends to eat earthworms. So you've got a few exceptions to this general pattern of snakes specializing on vertebrate prey. Now, varandas are an interesting case because these are basically a group of Australian lizards that's on the road to snakehood in a sense, where a appreciable fraction of their diet is vertebrate prey, but they also eat invertebrates and they eat carrion. So, varandas can actually be very opportunistic in terms of what they eat. So varandas are going into the vertebrate domain, but they still have attributes that keep them similar to other lizards in terms of diet. So let's look at this little batch of points right here. I want to draw your attention to one point here. Specializes on squamates. And that lizard turns out to be one of my favorite lizards, which is Lialis burtonus. So this is Burton's snake lizard. So this is actually a gecko. So this is one of Australia's massive radiation of legless geckos, about 50 species of legless geckos in Australia, pygopodids. And this species is essentially the snake among lizards. And so what Lialis actually specializes on are skinks. And so things like Tenotus in particular. And if you look at snakes, if you ever CT scanned, or if you look at skinks and you've ever CT scanned a skink, for example, you know that they have lots of osteoderms. In fact, skinks are actually quite hard for a lot of things to feed on. They have those osteoderms. It makes it really tough for things like birds to actually get a grip on them. So, and they're really good at rolling out of beaks and rolling away from other sorts of things that might grab them, including snakes. So a lot of things that feed on skinks have very specialized dentition and other sorts of adaptations. Well, Lialis is no exception. In fact, Lialis actually has this amazing hinge right in the center of its cranium that I like to think of as snake pliers. So here I've got a Lialis in my hand, and I'm just pressing against the front of its snout. And what you can see is it actually bends that cranial hinge. And what that basically lets Lialis do is wrap its jaws around skinks and basically increase the surface area, the contact area between its teeth and the skinks on which it's actually trying to get. And it actually has another adaptation as well. It actually has hinged teeth that basically let things go one way down its mouth, and it doesn't really let things back up. So there's really two major morphological specializations here that makes this essentially a skink super predator. So that's what your most snake-like lizard is, perhaps perhaps anywhere. So that's sort of looking at snakes versus lizards in some ways and some oddities, but what about how do lizard diets differ between these systems? So here I'm actually removing snakes, and I'm only keeping lizards in the picture here, traditional lizards. And so basically what I'm still coding, though, is things by Australia versus the Amazon. And interestingly, though, you can still see a strong signal of dietary differences between Australia and the Amazon. So despite the fact that these things are generally invertebrous, with few exceptions, if you look at how these things are actually factoring out, you tend to see Australia focusing on more termites and more ants in their diets, and you see within the Amazonian communities, you see the lizards eating more spiders and more insect larvae in particular. And so this is basically a pattern that's been known, some work by Laurie Vitt, collaborators, Eric Pianka, and others, and they basically looked at how neotropical lizards and desert lizards basically partition some of these major dietary categories with respect to clade. And so basically what you're seeing here is this essentially a phylogeny with diet matched in from some data from Laurie Vitt. And you're basically looking on the top at neotropical lizards, and you can see the percentage of termites that are in their diets within a couple of major clades in that tree. And you can see on the bottom the percentage of the diets for those same two clades that are termites in Australia. So much higher percentage of termites across the board in lizard diets in Australia. So just to sort of finish, what I want to focus on is what I think of as some big outstanding questions that I find really intriguing, things that we can't answer right now, and that I hope that maybe sometime with enough basic natural history data about these systems we'll actually make some progress on. First of all, why are there so many snakes in the neotropics? Why does the neotropics stand out as being the snakiest place on Earth, at least in terms of species diversity? So 69 to 70% snake in the western Peruvian Amazon, 17% snake in a typical community in arid Australia. Well, an obvious possibility here is maybe you have more niches of some sort. So one question might be, do neotropical snakes specialize on resources that are simply unavailable in arid Australia? After all, it doesn't take... you can sort of do a cursory examination of snake diversity and see, well, obviously there are lots of aquatic species and arboreal species, and there aren't as many trees or watery places in deserts. So very obvious sort of potential explanation. And in fact, if you look at that from that overall species abundance distribution that you're seeing, and now I'm pulling out the species that are aquatic and arboreal and showing you that, in fact, there are a lot of species that are aquatic and arboreal. These include things like helicops, Imantodes, Dipsis, Corallus, hydrops, and other sorts of things. So there's a broader range of habitats that's used in the Amazon. There's no question about that. But even if you remove the specialists in that sense and focus primarily on semi-fossorial and terrestrial snakes, excluding snakes that sleep in trees, but focus on snakes that spend their time hunting on the ground, like most Chironius, for example, you basically still end up with about 40% snake in the western Amazon in terms of 40% of that community is basically terrestrial and semi-fossorial snake versus about 17% in Australia. still have an excess of snakes to explain. Second, why are there so many actively foraging non-snakes in Australia? So now you're looking again at these are the overall species richness distributions for Australia on the left and the Amazon on the right. Now, if you look at just the clades, excluding snakes that are active foragers, using snake clades that use chemosensory prey detection systems, and they use their jaws for foraging, for prey capture, you have about 67% of that distribution are lizard species that fall into that category in Australia, versus only about 17% that actually fall into that category within the Amazonian community. So that's gymnophthalmids, geckos, and skinks in the neotropics. And in Australia, it's skinks, geckos, and varanids. So the diversity of these active foragers, excluding snakes, is much, much higher in Australia. So why? Well, one sort of superficial explanation might be that active foraging Australian clades are ecologically equivalent to neotropical snakes. But we've sort of looked at this earlier, and we can see that snakes actually fall out in a very different region of ecological space, at least trophically, relative to lizards in general. So this is, again, lizards and snakes, and these are neotropical taxa here, and what you can basically see is that, again, snakes and lizards are distinct in trophic space. And so this is not a very good explanation. So snakes are in a highly distinct region of trophic space. And finally, and this is one of the questions that keeps me up at night, why are there so many rare things in the neotropics? So Eric Pianka published this paper in 2013, and Eric's been working in Australia for a long time, has an insane amount of data on community ecology of Australian lizards in particular. And it's this really nice paper that summarizes in some ways his work in terms of thinking about causes of species rarity. So he looks at the distribution of species richness, with species abundance distributions in the Australian deserts, and says, starting from the first line of his abstract, most species of Australian desert lizards are uncommon, which, sort of follows from that species abundance distribution that is from his paper right there that you're seeing below. But when I look at this and I see, okay, you compare Australia to the neotropics in terms of species abundance distributions and detectability and so on, it's really, the Amazon really harbors things that are far rarer in general than you would have in Australia. So Australia is just far, even the rare things in Australia are much more common than the rare things in the Amazon. I don't know how many years you would actually have to take to essentially saturate out a local species accumulation curve in the Amazon, but I think it would be many, maybe lifetimes worth of work. And so I think that the consequence of this is that the effective rarity is actually much higher in the neotropics. Now, there's a lot of questions we might ask here. Are these animals really rare? Or is this just a function of detection probabilities being higher in Australia, so trappability, everything else being higher in Australia? Or is this, but nonetheless, though, I think there's still the consequence of effective rarity is that it's very challenging to understand diversity in these neotropical systems. Much harder in some ways than, we don't understand Australia yet, but I think it's even harder to understand the neotropics for these reasons. And so I get back to this sort of fundamental set of questions that I was posed about basic natural history of things like, Bothrops bilineatus, looking at, habitat usage and behavior and how they interact with predators and prey. And let's just take a couple of these questions that maybe we can think about some ways in which modern technologies can help us solve this knowledge gap for things like, for taxa like this. So if we focus on these questions about abundance, we might say, this is a case where maybe we can actually take some modern, slick tools of population genomics with some theory and try to say some things about species abundance. Though in principle, at the simplest level, genetic diversity is a function of effective population size and mutation rate. So there's got to be some way that we can actually do something with the patterns of genetic variation to actually say something about species abundance. Well, we've actually tested this with Australia, where we actually have decent abundance data on relative species abundance that I trust, more so than the neotropical data. And so we looked at this whether genomic variation, so using sort of broad scale surveys of genomic variation across 5,000 loci for lots of individual lizards spanning rare and common species within Australia, actually tracked what we could estimate in terms of census population sizes from field surveys. And so overall, we found that, yes, species abundance does correlate with, as we measure it ecologically from field surveys, species abundance does correlate with standing genetic variation. So doing some things to, try to think about how non-equilibrium demography would mess with it. Nonetheless, there's this positive correlation, but it doesn't explain very much of the variation. It might be about 15% of the variation in abundance, 15% of the variation in genetic diversity is explained by abundance. And I don't want to call it depressing, but the message that we actually have in this paper is that we conclude that genetic diversity does no better at predicting abundance than a single day of traditional sampling in this community. That means, so we did a number of analyses to ask, if you go to these communities in Australia and you do one day of pitfall trapping, does that let you predict your long-term abundance estimates better than this fancy genomic approach? And the answer is no. So it basically works out equal. So one day of traditional sampling is all the fancy genomics that you can do in terms of abundance reconstruction. So I think there's some messages here about understanding tropical reptile diversity, and I think we really face serious challenges in understanding, especially the ecology and natural history of tropical reptile diversity. And so this is sort of how I think about understanding reptile diversity in terms of cost per unit of per some unit of biodiversity or natural history knowledge. And so on the x-axis, you've got how many dollars are we spending to understand something about ecology or natural history of a system, species diversity, whatever. On the y-axis, you've got the return on that investment. Basically, how much biodiversity data do you get, whether it's specimens or tissues or ecological time series, whatever. Now, I like to contrast this to the disdain of ichthyologists with fish, because I think fish are a good counterpoint to what I think the situation is for reptiles. So let's say you're interested in fish ecology. So I think the curve for fish looks like this, an exponentially growing curve where the more you spend, proportionately, the more you get. It's kind of like people who are very wealthy can get even wealthier because they can hire people to do tax tricks for them and they can make slick investments and get even wealthier, right? We know this phenomenon happens in terms of the economy of human societies. I would argue that it happens in some ways with biodiversity knowledge and maybe with groups like fish. What do I mean? Well, let's say you have a little bit of money. Well, you can go down to the store and buy a small net, collect fish in a local stream. You have a little bit more money, you can buy a bigger net. You don't buy you don't have two people with little nets, you buy a big net. And if you have even more money, you can buy an electroshocking backpack like this, and then maybe you can actually buy a boat. And then if you have lots and lots of money, like you can buy a really, really big boat. So you can actually go and seine things and trawl and everything else. So I would argue that the more money you have, proportionately more you get in terms of biodiversity data. I would say that, especially for neotropical squamates, though, I would say that the curve looks a lot more like this. And really, you have a near linear increase in investment, where the only thing we're still stuck with people putting pitfall traps in the ground, checking pitfall traps, going out at night and looking for things. Can't think of acoustic monitoring systems that are going to work for squamates in general. And so we really don't have any game-changing technologies. We've tried to look at genomics. Maybe this will be a salvation or a shortcut here. It doesn't seem to work as well as it's cool, but it's kind of an exercise in what we can know in principle, but doesn't seem to be a game-changing technology. And so I would say that I think that basic natural history data is critical for all these kinds of questions that I'm interested in terms of overall species diversity from a macroevolutionary or macroecological perspective, and I think continued fieldwork is fundamental. That's actually one of the really exciting things that I see about this conference here, because I think actually at this conference in particular, I interact with a lot of people who really value this kind of basic natural history data about what things do in nature. And so I guess I'll finish there, and there's lots of folks to thank for this work, and I thank in particular, although I definitely can't name them, the literally hundreds of individuals over the past 17 or so years or 15 years who basically helped me with field work, both here in the Amazon as well as in Australia, from government agencies to Aboriginal groups and so on. So thank you for your time.